Welcome to the first day, the first full day of didactics for the course. I think last night was a really successful and useful set of sessions. The one thing I'm going to try to go over what we're hoping to accomplish today, and I'll just do that by briefly giving you an overview of the agenda today. Right now, I'm giving an introduction to the course. The thing that I think I want to just stress is again, I talked about this a little last night. The clarity of purpose here is that we want you guys to move ahead your protocols as much as possible while here, and also take advantage of the others in your small group to learn and get to know them, and to help give them feedback on their protocols, including both your faculty and the other trainees that are in your groups. We think that the more looks that your protocol gets while you're here, the farther along it will move scientifically. And also, don't hesitate to reach out to other faculty or trainees who are here, who are in your area, who could provide advice. We have lists of the faculty in their areas in the in your folders. After this introduction, which I hopefully don't take too long doing, Chris Coffey is going to give this excellent overview of clinical trials, and then Sharon Yates is going to be talking about what information you need before talking to your biostatistician about sample size, and then we'll have a short break. Karen Johnston will then be talking about developing a schedule of events for a clinical trial. So a couple things of note to be cognizant of is a couple of the small group sessions are mostly in other rooms. 2 of the small groups are meeting in here. And that will mean that when the sessions are done, it will be important that if you aren't one of the 2 small groups that are meeting in here, that you, I guess to be blunt, get out of here as quickly as possible. So, so we're not cutting into their time, so that they can get started with their things. In addition, one of the things that we want you to do is provide us feedback on these sessions so that we can make them better in the future. There's an evaluation link up at the top, bit.ly/A2 course. And then on the back, so these evaluations are anonymous because we want anonymous feedback. We want people to be frank. And for those of you who need CME, CME is available for them. You need to email us the total number of hours that you attended at the end. But in order to keep track of which thing, one of the things is there's a lot of sessions, so you fill out a lot of evaluations. And you might lose track of all of the evaluation. Did I evaluate that session? Did I not evaluate that session? So on the back of your agenda, there's a checklist. And you say, well, wasn't there a more electronically sophisticated way that you could have done that? And I don't know, how many of you have electronic health records at your health systems? Did the people who designed those electronic health records ever look and see, is what we're designing as good as pen and paper was? And I would say that they probably didn't. So so anyway, that's how you keep track of it. I would suggest you put your name on your agenda so you know which ones you've evaluated. But again, it's very important for us to get your feedback on the sessions. So then after Karen's session, when you move into the small groups, you're going to be working on developing a schedule of event for a clinical trial. And the goal of that session with you and your faculty is to look through and actually do the exercise for your clinical trials. And you may not get through all 4 of them, but the idea is to take the sort of lowest hanging fruit and work on that. Then we'll be moving into lunch, and then after that, we will be going on to a couple more didactic sessions. Jeremy Schefter will be presenting on selecting outcome measures, and then Dr. Ken Chung will be presenting on learn phase designs and the CRM. After a short break, Valerie Stevenson from the Neurologic Emergency Use Treatment Trials Network will be presenting on creating an effective clinical trial budget. And then each of the 8 small groups will be breaking up into their own groups, and you'll have an exercise which has sort of 4 different scenarios, and based on what you've learned from Valerie's lecture, each of the small groups is going to work on coming up with a budget for each of those sessions, for each of those examples. And then I'll come back to the large group and then discuss the different budgets we arrived at. And I think it'll be interesting to see what people come up with and what the variability is. We'll have, after that discussion, the formal agenda for today will be closed. What we are encouraging all of the small groups to do today is try to have dinner together and also work on their plans for what needs to be accomplished on the protocols throughout the rest of the week. We were discussing briefly, people were maybe thought that maybe I was unaware that that Ann Arbor is a cool town, and I have you trapped in a suburban office park, which I'm aware of. But there's these things called taxis and Uber, and if you don't have an Uber app, you can ask if you already are in Uber, one of the other people in Uber can send you a code and you get $20 of free Uber rides. Obviously this is a pyramid scheme, but if you can't find somebody that has a Uber discount code, I just feel a little bit conflicted because then I'm getting, when somebody refers you, you get $20, they get $20. So again, it may make more sense for your actual faculty member to do it, so that way you guys get a ride there and a ride back. Uber seems very safe and reasonable in Ann Arbor. The last time I wrote it, it was totally fine. So Ann Arbor is a very safe place. Tomorrow we're going to have a large group dinner all together in downtown Ann Arbor, and we will all be together for that. We'll have shuttles up there, and that has been graciously sponsored by the American Academy of Neurology. So I think you should give the American Academy of Neurology a hand. And we do, and there isn't a misprint. I do intend to go to karaoke after that. So, a show of hands, maybe, as to who would go, because I do need to make a reservation to karaoke. Or maybe I should send out an anonymous survey. I'll send out an anonymous survey. Just check your email. So not everybody. There's no requirement that people have to participate, but you're just watching me. The rooms are small enough that not everybody would have the opportunity to watch me. So I'm sure somebody will memorialize it on video. With that, I think, let's see. Oh, we have 2 minutes. Are there any questions? Lori, Robin, did I miss anything? Okay, very good. All right, without further ado, why don't we start a minute or two early? I would like to introduce one of the course directors, Chris Coffey from the University of Iowa, who's going to be talking about an overview of randomized clinical trials.
So when you said it can watch you tomorrow night, does that watch you whip, watch you nay-nay? What? Does anyone get that? You never know what might happen. All right. So this is just kind of an introductory overview of clinical trials. So a lot of what I'm going to say is probably reviewed for many of you, but I think it helps to kind of make sure we're starting off kind of on this thing, general ground for some of the basic stuff, which some of this is also probably come up in the small groups. I mean, one of the key things that that repeatedly comes up, and I think as you work on clinical trials is both good and bad, is these little blurbs. Now that we have this 24-hour news cycle, something happens, you see this little blurb either on TV, in the newspaper, or on the internet, something that comes out, which is all kind of the results of clinical trials. Interestingly, it's sometimes interesting to read the actual paper and then read the press release and see how they relate to each other. But it all kind of relates to a lot of the work that goes on in these types of clinical trials that we'll be talking about with the protocols that you're working on today. So what I want to talk about is spend a little bit of time just giving kind of a historical perspective. Okay, how did we get to where we are in the realm of clinical trials? Talk a little bit about terminology, phases, levels of evidence, and then towards the end talk about why randomized, kind of walk through a good example that kind of illustrates, I think, really well why randomization is really important in clinical trials, and then at the end just talk about key elements of a randomized clinical trial protocol, which you either are or will become very familiar with as you flesh out your individual protocols. So if you look at clinical trials, if you read kind of clinical trial history books, you can almost go back to So to the Bible, if you want to look at kind of the first, not clinical trial in the sense of how we would think of them today, but kind of a clinical trial as a comparison of different types of interventions. So this goes back into King Nebuchadnezzar, and Babylon came under control and ordered everyone, under his order to only eat meat and only drink wine. And so there's this story about some of the this one group who wanted to eat vegetables and drink water, and so the king, didn't think that was a good idea, and said, okay, you can do this for 10 days. Right? You can eat this diet for 10 days. We'll see how you do. Okay. So then after the end of 10 days, the group that was drinking water, eating vegetables, looked fitter than the group that was sitting around eating meat, drinking wine all the time. So they said, okay, we'll let you continue to do this diet. So it wasn't randomized, none of that, but it's kind of an interesting, kind of in general aspect of the types of things that we're looking at in clinical trials, kind of comparing one intervention to another. When you get into really things more recently that lead into a lot of what we look at. So in 1537, Meurer, so this is kind of an unintentional trial. So this happened almost by accident. So this was in a battle in France where he was a physician who was treating soldiers who were wounded. And at the time, the standard of treatment if you were wounded in battle was you would pour boiling oil over the wound in order to, quote, heal the wound. And so in this battle, they were running out of boiling oil, so they needed to come up with an alternate strategy. So they made up this other treatment from egg yolks, turpentine, oil of roses that they could rub on the wounds. And what they found was the people who they put this turpentine type mixture on their wounds healed better than those they poured oil on, right? So it's kind of an interesting dynamic in a lot of different ways. It's not clear whether the turpentine mixture actually worked, or whether it was just better than pouring boiling oil. But if you go on, so famous example, which many of you probably heard of, the scurvy study. So this is again soldiers who developed scurvy on a battleship, and so at the time, again, there were a lot of weird treatments when you read back kind of the history of medicine. So at the time, if you had scurvy on a ship, that you would drink seawater. That was thought to cure you from scurvy. So they tried different things. They looked at a lot of different types of combinations of things to treat scurvy. The one that worked the best, which makes perfect sense now, was a mixture of oranges and lemons. And so they realized that was, the soldiers with scurvy that had the oranges and lemons were recovering better than those who drank seawater or the various other things they had in here. So when you get more recently, 1863, so there was a rheumatism study, which is kind of the first placebo-controlled trial. So this was a population of rheumatic fever patients, and the treatment here was mint water. So they were really just giving them mint water and comparing to previously described effects. And so what they found, there was really no difference that they were seeing. So the effect, so they had done individual studies with the treatment that wasn't compared to anything and found some benefits. They did a study where they just gave them mint water or a placebo and found similar effects. So it's kind of the first study that documented, what we commonly refer to as the placebo effects in trials, which is an important area that you have to consider if you're trying to show that a treatment is better in terms of giving some outcomes. And in some disease areas in our center, we're working on a pediatric migraine study where the placebo effect is as much as 50%, right? So half the people who you do nothing to get better over time. So if that's the scenario that you're in your type of protocol that you're working on, you're going to have a treatment that works really, really well because it's got to work better than this underlying placebo effect that is in the background. So 1923 is really the first I've been able to find the first application of randomization, which comes from R.A. Fisher, who, in the statistical world, everything kind of comes back to Fisher. He worked on a lot of agricultural type research. So this is not necessarily in the clinical trials setting. This is more in crops and looking at different plots of land and randomizing plots of land to different fertilizers to try to see what the optimal combination of fertilizer was to yield the most gains in terms of the crops. In 1943, so this was a study done out of the UK. It is often known as the first double-blind control trial. So this was a trial that was done in a British, this was British office and factory workers who were suffering from a cold. And so there was Patulin, which was the intervention versus placebo. And what they did here was an alternate procedure. So it wasn't Again, this wasn't randomized the way that we think of it today. What they did is they said the first person came in and got treatment, the next person got placebo, the third person got treatment and placebo. So it was, not random in the sense that it was random, but it was better that they at least rotated individuals. They didn't show any protective effect. It's kind of interesting. It's the first attempt at randomization. And when you get into actually the first randomized controlled trial that used a randomization procedure, that came a few years later again in a study out of the UK with streptomycin, where they compared it to placebo. And what they were able to, this was actually a very interesting study when you go back and look at the history of clinical trials. It was the first double blind, but it has a lot of interesting facets. So they showed that streptomycin was beneficial, but in the long-term follow-up, they also showed that it was beneficial for short-term, but eventually the patients would develop a resistance to it. So it had some benefit, but not long-term benefit. So kind of an interesting case study in terms of the history of clinical trials and what was done. And a lot of what we do today kind of reaches back to a lot of things that were discovered in that study. I pulled up here some other kind of key dates in the history of clinical trials, which is just kind of good to know from a historical perspective. So 1906 was when Congress passed the Food and Drug Act. So before that, there was really no regulation of any of this. And that was where you see like the old movies or TV shows where you had people who would drive around from town to town, the doctors who would sell their potions, out of bottles, and all that was perfectly fair game. So when FDA was first formed, they didn't quite have the teeth to regulate it. That kind of came later, and then there's been other standards in terms of human subject protections that have evolved over time. 1947 was the Nuremberg codes, and that came out of World War II and a lot of the experiments that were done in Nazi Germany on prisoners or people who had not given consent. So a lot of the language about informed consent that came from that you can't just put somebody in a study without them understanding what you're doing to them, why they're doing it, and what you might gain from it, and so forth. The Belmont Report, the Declaration of Helsinki in '64, the Belmont Report in '79, and all these things. And a lot of this seems intuitive, but you look and there's actually, there's interesting history, even in recent years, of things where you have the needs or the wants of science sometimes conflict with the needs or wants of the individual, and that's a paradigm that's often a struggle in terms of how do you address that. Even in the U.S., we think we would never do the types of things that they did in Nazi Germany and different things like that. You have things like Tuskegee, right, which is a horrible situation where it wasn't that it was originally not consented. So they were enrolled in a syphilis study. It was just as penicillin was developed as a treatment for syphilis, it was withheld from the people in the study. They were never informed about it because, again, it was for scientific reasons. The goal of the study was to look at the natural history of syphilis. And obviously, if you give a treatment, you can't look at the natural history because you've modified the natural history, but that's really not an ethical reason to withhold something like that. And we run into that today. We struggle with that. If you're working in a disease area where maybe a treatment comes out, it's not the perfect treatment, that your patients might go on that. Now, if you're looking at something that's beneficial, it needs to have an effect above and beyond that treatment effect, right? So you have to examine the impact there. Some more recent things. So there's some ICH, good clinical practice. There's a lot of FDA guidance documents, ICH documents that you should make yourself familiar with in the areas of what you're working with. 2006 with the CTSA awards, that's really changed kind of the way the interaction and works between the translational side and the clinical side. And this helped to make that more efficient. And then in 2009, with the Recovery Act, there was money allocated for comparative effectiveness type research. Now, I'll come back to that in a minute because that's an area I think that is that's getting more and more attention in recent years. So hopefully this is something, since you're at the course, you're familiar with this, but the definitions in here just to reiterate. So a clinical trial is just an experiment that's testing medical treatments or prevention strategies on human subjects, right? So it's involving the administration of some test treatment. A randomized clinical trial is doing that with at least one control and at least one test treatment, right? You might have more than one test, you might have more than one control in some situations. So you're doing this in a randomized fashion. And some things that you have to consider here. So there's blinding or masking, which will be extremely important for you to consider as you're working on your protocol. I'll confess, I'll call it blinding most of the time because that's what I do. There are some areas where masking is a preferred term. If you work on studies with the National Eye Institute, for example, blinding is kind of a sensitive term, so masking is probably there. So I've tried over the years to make myself use masking, and it's like, old habits die hard. But there's different types. There's single blind, there's double blind, there's triple blind. Right? I think so. I'll give you the official policy, and then I'll give you my opinions. Right. So the policy is blinding is important, right, to ensure the integrity. My personal bias is I don't know in a lot of situations it makes that big of a difference. We're doing it kind of to eliminate perception as much as reality, right? There are situations where it can't, right? And there are things where it's really hard to blind. If you're doing a surgical study, it's hard to blind a surgical study. If you're randomizing to get surgery to not get surgery, then it's how do you blind that? You have to have someone go in, they think they're doing surgery, you don't really cut into them, or maybe you cut into them so they've got a scar that has to. There's different levels of how far you go, how far you can go. But in situations like that, if you can't blind the intervention, you want to blind the outcome assessment. And this gets into some of the outcome type situations. You want to have someone who's independent of the whole procedure, do the assessment of the outcome, and evaluate the patient, if it's a 30-day endpoint, independent of knowing whether they had the surgery or not. And there's other things, and this is where I think blinding will become more important when you get into studies like what was addressed last night in patient-reported outcomes. If you're doing patient-reported outcomes, blinding of the patient is going to be really important, because if I'm a patient and I know I'm on an active treatment versus placebo, whether consciously or subconsciously, that's got to affect how I feel like I'm going to respond. Maybe if I'm filling out a headache diary and I know I'm on active treatment, maybe I feel like, well, if you think this is not a headache today, versus if I'm on placebo, maybe I say, yeah, this is probably a headache today, right? There are ways that can impact you, whether it's conscious or not. It's also important to blind the investigator. To a couple of things, right? One of the things that comes up in discussions, statistician working with clinicians are blinded to treatment, but also physicians typically need to be blinded to results while the study's going on. So this is very different than other areas of science where you're doing a study and the professional meeting is coming up, so the study's not done, but you want to run interim stuff to try to get an abstract so you can present at the meeting. That's usually not appropriate in clinical trials, because as the investigator, if you kind of have a sense of how the study is going, that can potentially impact how you manage the study, how you determine who to encourage your investigators to enroll in the study and so forth. Typically, as an investigator, you're blinded to the results. Even if there's interim analyses or interim plans, that's usually not the investigator of the trial who is open to that. That would be your oversight boards. If you would only be brought in, typically, if one of two things happens, either there's really good results and the study might stop early for benefit, or something really bad is happening where you either have to make a major protocol change because there's a safety issue, or the trial may be shut down. And I think there's talk tomorrow, Will, about the UNC, is that right, on Tuesday? That gets into more of that aspect. Now, the other things that are important to consider are the types of treatments. A lot of the areas where I work on, and probably a lot of what you're working on, are drug type of interventions, but those are not the only types of treatments that you can have. I mentioned surgical procedures, there's devices, there's different kind of therapeutic modalities, there's diets, behavioral intervention, exercise regimens. So there's any type of treatment that you can have. It doesn't have to be a drug. In terms of controls, we'll often talk about placebo-controlled trials, although again, it doesn't always have to have placebo as the control group. So if you're working in an area where there is an active treatment that works in that standard of care, it would be very difficult to do a placebo-controlled trial because you can't put someone in your control group at kind of worse than what they would get as standard of care. So it might be an active control. It might be looking at combination therapy versus single therapy. There's lots of different ways that can happen. You may be doing something with a non-inferiority study. Are you guys familiar with non-inferiority studies, right? Where if you think there's an active agent, maybe it works, but it's got a safety profile that's questionable. If you have a drug that works as well but has a better safety profile, that might be an attractive type of feature. So it really depends on the study and what the main objective of the study is for you to do. I'll talk a little bit about the phases of research, although this is kind of more the traditional way it's laid out. In a lot of modern clinical trials, there's a lot more blurring of these lines than there used to be. So you could think of it in terms of the separate phases, or you could think of it broadly. There's also language you'll see as learn phase and confirm phase, where in particular a lot of the early phase areas are kind of morphed together depending upon the objectives and the goals of an individual study. But I think just to help maybe put it in a general sense of what the goals are, there's phase one, which are really mostly focused on safety and toxicity profile. These are typically the first in humans, small, uncontrolled types of studies. So a lot of these are that's the first time that you're looking at it and you're trying to see how high can we go with dosing the drug before really bad things happen. And we just had a group of summer biostat students who were visiting Iowa, and they came and I talked to them. These are the studies that they're the kids who are doing this. This is where you walk around, like, you go down to the University of Michigan campus and there's flyers, right? Want to make $200, $300 over the weekend. So you go in and give you the drug, they take your blood every hour and say, see what happens to you. So in some sense, as much as any type of trial, this is the guinea pig, right? These are usually healthy volunteers who have really nothing to gain and potentially a lot to lose from this. But so you're often reimbursing them financially because you're trying to get a sense of what the drug does in the system. And you want to get a sense of that typically in a healthy population before you go into a disease population. And then once you've done that, you have a sense of where you can go safely. Then you move more into the proof of concept type of phase, which is more the phase two, where now you're looking at people with the disease or the area that you're wanting to treat, and you're trying to see, right, is it safe, right? You're still looking at safety. Safety's always underlined there. You're really looking at more of this go, no-go type decision. So you can't, a couple of important points in phase two. You can't show that a drug is safe. I think over the years I've worked with tons of investigators who they wanted aim for their trial to be, we want to show that the drug is safe. Can't do that, right? Can't do that in a large phase 3 trial. You can show that it's unsafe, but you can't show that it's safe. You can only rule out, some level of safety concerns based on the size of your study. You can't show that a drug is efficacious in terms of your main clinical outcome in phase 2, or else you're basically doing the phase 3 study. And if you're doing just a mini version of the phase 3 study, you're kind of doing an underpowered phase 3 study with the idea already made, and this go, no-go criteria, you're going forward no matter what, because you don't really have the power to address the question of interest. So a lot of times what you're doing here is you're trying to set up some threshold to see, I need to look at and see whether there's enough evidence of an effect here that it would justify moving forward into the next phase, the phase 3, the more confirmatory phase, which is much more expensive, much more time-consuming, much more costly, both in terms of your time and effort, patient time and effort, and so forth. Is it worthwhile to do that? So that's the goal of phase 2. I think in general, as the statistician, I'll say, and unfortunately this is where a lot of you probably live in the protocols you're developing, because you're not going to jump into being PI of a large phase 3 trial as your first trial. But phase 2 are some of the hardest to design because you're not doing an efficacy study per se. You're trying to come up with this go, no-go criteria, and you're trying to balance the resources that can be available for that phase 2 trial with a question that you can adequately answer and get a sense of whether you're looking at something that's promising or not. The phase 3 that I alluded to, that's kind of what you typically think about, and that's what we hear in the news more, the large confirmatory studies, which you're looking at really to try to get a sense of whether what you're looking at actually works. And this is where you have to have typically 2 of these for an FDA type of submission. A lot of criticism in recent years, and there's been a lot of attention on this, is that we should spend more time in phase 2. There's often a rush to get to phase 3, right? That comes from a lot of different resources. There's a lot of competing things. A lot of times, the patient communities, like we heard last night, want to see things move through quickly. But there's also danger to going to phase 3 too early, right? If you don't know dose, you don't know, what type of criteria to do, you don't have a sense of how to power the study for phase 3. You might go to phase 3 and fail, not necessarily because the treatment didn't work, but because you really didn't have all your eggs in a basket before you went to phase 3. And I think one of the things that you can argue is perhaps I don't know if the most unethical thing is the right term, that might be a little too strong, but one of the most dangerous things that you can do is an underpowered phase 3 confirmatory study, because you might have a drug that works, and you do an underpowered phase 3, it's, quote, negative, right? When it's reported in the press, a negative study means the drug didn't work, whereas negative, in my sense, means you didn't answer the question. If the drug didn't work, and you find that the drug didn't work, that's a positive study. But if you had a study that showed the drug didn't work because you did not have adequate power to show it worked, if it actually had an effect of interest, that's a negative study, right? And that's the worst case situation, because then that drug's going to go on the shelf. No one's going to touch that because you just wasted millions of dollars in a phase 3 clinical trial that didn't show benefit of the drug. And it's not going to get looked at anymore. Whereas if you spent more time, had a better sense of that, and did an adequately powered study, maybe you would have been able to show benefit with respect to that drug. Or you don't show benefit, but it's clear that you're not showing benefit because most likely it doesn't work, and you answered the question. That, at least from a statistical perspective, is a positive study. It's negative from the patient community perspective, right? Because the drug doesn't work, it's not going to be beneficial to them. But from a scientific perspective, it's positive if you answered the question. There's also phase 4 studies, which are these post-marketing surveillance studies. You heard a little bit about those last night. Again, this is really to get more at the safety aspects, because you can't show, even in the large phase 3 trials, you're not going to show safety. Right? You may have thousands of people in your phase 3 studies, but if you have a drug that maybe something really bad happens in 1 in 10,000 people, you may or may not see that at all in your phase 3 trials. But if that drug is approved and then goes out and millions of people take the drug, you're going to start to see those problems creep up. So that's why this long-term surveillance is important. The other thing that's becoming, I think, more prevalent and, is comparative effectiveness trials. I mentioned this a little earlier. So what this gets in, there's been a focus in recent years on this. And this gets a lot back to kind of the academic investigators and the CTSAs and all are involved in this. And this is if you have, this is where you start to mix science and policy, really. That if you've got a disease and say there's 3 treatments that are approved to treat something, then as a physician, if someone comes to you, you got to make a decision on which drug to put that patient on, right? So comparative effectiveness are really based on studies to try to make educated decisions, right? Evidence-based types of decisions on which drug to put someone on if they show up in your clinic. The challenge with these studies traditionally is if you've got 3 approved drugs from 3 different drug companies, it's really not in anyone's best interest to fund that study, right? Because if there is one winner, at the end of the day, somebody's going to be happy and 2 people are going to be unhappy, and it's going to affect, lots of downstream implications. So that's really shifted more in terms of, NIH is focused on this. That was a lot of the Recovery Act money to fund more of these comparative effectiveness type trials to try to get answers head to head. Are there things that if someone shows up in the clinic with the disease, what's the first-line treatment, best first-line treatment to put them on? What's the best, kind of combination of treatments, things to put someone on? So really getting back to questions, I think that as a clinician in practice is probably interesting to you, that you want to know in terms of how to treat, an individual patient, that really we haven't addressed much in the clinical trials realm because it's been more in drug development and getting approvals and doing the head-to-head comparisons after approvals hasn't really been high on the radar of priorities. A couple of other things that are worth mentioning. So there's levels of evidence, which I think you guys are probably mostly familiar with here, where you're looking at, so you get like level 1, level 2, level 3. There's also class A, class B, class C, which is all based on, is it high quality, low quality? Is it multiple clinical trials? Is it one clinical trial? Is it more observational type evidence? Which gets into more of the policy type issues. What are standards of treatment? What are the recommendations to support those, and so forth? In terms of why randomized, so here the goal here really is to come up with groups that are comparable, right, in terms of characteristics. So in theory, with randomization, randomization should lead to balanced groups on all measured and unmeasured characteristics. So the only differences are what you're randomizing to, and that any differences you see are related to that. Obviously, if you're in smaller settings, that doesn't work well, but in theory, that's what you're looking at. I think a good example of where randomization is kind of important, even when we think we know the answer, and this is a bit dated example now, but I think it's still a really good example, is the Women's Health Initiative. So if you look at the Women's Health Initiative, so this is, I pulled this from a New England Journal of Medicine article from 1991, where there were lots of benefits. So at the time, in the early 90s, there was this whole push with hormone replacement therapy, and it was thought to be lots of benefits to hormone replacement therapy. If you're postmenopausal women, it was, most postmenopausal women at the time were going on hormone replacement therapy because of these perceived health benefits. And here's like the cover of Time magazine in June of 1995, where there's this whole article that's really kind of talking about the prevailing medical view. In essence, at the time, you should go on hormone replacement therapy. the condensed version. If you're a woman, there were lots of beneficial risks. So around this time is when kind of the Women's Health Initiative was started, which is kind of an interesting case study for a lot of different areas in clinical trials. It had, it had politics, it had lots of different areas. So it was the largest, probably is still the largest, I'm not really sure, but I would guess, study ever funded by the federal government exclusively in women, right? So it had a lot of political appeal for that, because then, politicians could go back to their districts and say, yes, I helped the vote to get the study through. But it also had different components. There was a nutrition component, but there was this hormone replacement therapy component, which was one of the areas to it, which was actually somewhat controversial of whether it should be done. And so there's this whole issue of equipoise in clinical trials, which is also important for you to consider in your protocol. And the equipoise issue gets to, if you know that one treatment is better than the other, you should do a randomized clinical trial, right? It would be unethical to randomize people to one of two interventions if you know that one is better than the other. So equipoise is this idea that you don't know, right? Maybe your new treatment could be better, maybe it could be worse, maybe it doesn't work, but you feel like that there's some level of equipoise. Where that is, it's usually more of a societal level of equipoise, because I've never worked with an investigator who wasn't convinced that the drug that they're developing and have all this preliminary data on works, but you're looking at more at a societal level, your colleagues, is there equipoise out there? There was a lot of concerns in the Women's Health Initiative being formed of whether there was actually equipoise to do this, because a lot of the community, as evidenced by this Time magazine article, was that hormone replacement therapy worked and was beneficial. And nevertheless, because a lot of the different components I mentioned, it was done. And what actually happened was a bit surprising. So they actually stopped the study early because there were some health risks that were observed that really were not expected at all when the study was launched. And so you saw this shift in time from, so this is the June 95 cover of Time, and if you go forward, here's the 2002. So it kind of completely changed the perspective. And there's been a lot written about the Women's Health Initiative since, and a lot of things looking at it are the specific types of the hormone replacement therapy that are more beneficial than others. But it really is, I think, a good example, as I mentioned before, of where randomization is important. And one of the things I've read that I think probably has the most credence is one of the challenges, if you look at a lot of the observational studies that were done like in the 90s, is there was definitely a difference at the time of who took hormone replacement therapy that was correlated highly with socioeconomic status. So typically women of higher socioeconomic status were more likely to be taking hormone replacement therapy than women with lower socioeconomic status. And socioeconomic status is correlated with a lot of different health outcomes. Whether that's confounding some of the results of earlier studies is argumentative, but it's one theory that's out there. There's a lot of different discussion of that, but it's clear that sometimes even when we think we know something, we often get into the trial and realize that we don't know it. And I'm sure that most of the mentors on every team have their war stories of areas where they got into a clinical trial and ran into something that you totally did not expect, at the outset of the study and had to completely react to. It's kind of a standard that you kind of get used to. I like to describe clinical trials as it's organized chaos, because you never know what you're going to run into, but it has to have some sense of organization, and you have to keep things moving forward and address the things as they come up. I mentioned the equipoise issue, when to randomize. The thing that I like to put out there, and I try to use this kind of individually as well, is what I call the grandmother test, right? If you're doing a clinical trial, and you're enrolling patients in your clinic, if your grandmother came in with what you're trying to treat, could you look your grandmother in the eye and say, I think you should enroll in this trial, right? If you can't, then you probably shouldn't be enrolling somebody else's grandmother or mother or father or brother into the study, right? So I think that's an important thing to keep in mind as well. And that's where, at least individually, I think the equipoise issue has to come on. If you're running a study and you wouldn't put one of your family members in the study if they had this disease, it's hard to then justify how you would put somebody else's family members in, right? So that's where you get into this whole equipoise type issue and how do you justify the trial. And equipoise can shift. So you guys are not getting into the whole adaptive piece, that's more in the advanced track, but a lot of the language around adaptive designs, which you will hear if you get involved in clinical trials in your careers, tends with this, right? Equipoise can shift during a study. If you're deep into a trial and it's becoming clear that one works or not, how do you know when to make the decision to stop, right? When should you not continue randomizing people should you make adjustments to how likely someone is to get a drug or placebo. There's a lot of literature on that, lots of debate, and the sort of ethicists come in at the clinical trials and how do you relate all of that. So a couple of things that I want to address just here at the very end, at least kind of into the protocol. And this is the most important one, I think, here is what is the study question? So as a clinician, and what I'm going to say is probably going to be shocking to you, but as clinicians, you think different than we statisticians think. I know, good surprise, right? But a lot of the protocol development is trying to find a common language, right? So as a clinician, you come in with a goal clinically of what you want to do in the trial. And a lot of the early dialogue, most likely, I'm guessing the first maybe session, maybe first 2 sessions of your small group included a lot of this, is the statistician in the group trying to then say, okay, this is what you want to do. Let's put that in a testable hypothesis, right? How do I take the words you're describing clinically and put it into something that I can test statistically, right? And that's kind of the important aspect of the trial, is breaking it down from this conceptual clinical issue to what is it statistically that you want to test. What's the key question, right, that the study is trying to address? Because that then drives everything. The budget, the sample size, everything of the trial should be based on helping you answer that primary question. Because again, remember what I said just a few minutes ago. A positive trial is a trial that answers the question of interest. You know, if your question of interest is, my drug, say you're doing stroke, and my drug improves the mean, or improves the percentage of patients who get Rankin score 0 or 1 at 90 days, right? I can test that. At the end, I can see whether it did or it didn't, and I can determine the sample size I need to determine whether it did or didn't based on some specified effective interest, right? If I can do that, I can answer that question. It's a positive trial. If I go in and I just say, I want to see whether stroke can improve the Rankin score, right? That's a little different, and that's where you get into the primary endpoint, right? Saying, I want to see whether my treatment can improve the Rankin score. There's lots of different ways you can see an improvement in Rankin scores. The board knows the stroke literature is full of different discussions on what the best approach is. But if you haven't defined that, then you don't really know, so as a statistician, I don't know what to target for sample size, I don't know what your question is. So you have to take it a step further from just, I want to decrease the stroke rate. What do you mean by that, right? How are you going to measure that in your study? Is that looking at like a 0-1 for a Rankin outcome? Is that looking at like a shift score? Is that looking at a mean Rankin score? There's lots of different ways that you can compare that. It's important to take it all those different steps, right? So you take this broad question, and at each step, you kind of give it a little more specificity. What's the question? What's the endpoint, right? What's the hypothesis? What's the endpoint? How am I going to collect that? How am I going to measure that? And then the other important thing is, what am I going to do with missing data? Okay? Because if you have, in clinical trials, we have this concept called intent to treat, right? Which means, in basic terms, as randomized, if randomized, then analyzed. So everyone who's randomized in the study needs to be accounted for in the analysis. So, and that's for important reasons. So let's say I can't get a 90-day Rankin score because a third of the patients on my intervention group died, right? Then if I just ignore that and compare the mean Rankin scores, that's a really biased estimate, right? Because I'm not accounting for the fact that a lot of the people who I didn't get scores on had this really bad outcome that happened. I didn't get the score because they had this really bad outcome. So you have to account for that some way. So your protocol needs to have a plan for how you're going to address with missing data. Another thing that's tricky where you get informally missing data is when you have some type of cognitive type of endpoint. And so one of the things I've encountered in a couple of studies is if someone, maybe they don't have dementia, but they're starting to show cognitive decline. I've seen situations where you come into a visit and maybe you're supposed to get the cognitive testing. If you're cognitively impaired but not way cognitively impaired, you may not want to do that test because you know you're going to be fraud and you don't want to be reminded that you're having cognitive issues, so you just don't do that. Well, that's a missing piece of information, but from an analysis standpoint, that's an informatively missing piece of information, right? You did not get it because you're doing worse. So not including that is going to lead to a biased analysis. So you have to have some way to address for all of this. All this is tied into how you're going to address the endpoint, how you're going to answer the question of interest. So that has to drive everything in terms of the study. It really, other things are important operationally, but all those things don't matter if you don't have a question. If you don't have a question, you really don't have a trial. So you could have drug, you could have sites, you could have access to patients, but if you don't have a question, then it's not clear where you're going with the study. Inclusion-exclusion criteria, that's important. You'll deal a lot with that, and there's trade-offs here versus being less inclusive, being more inclusive. I think I'll tell you again, just based on experience, one of the common mistakes that I see new investigators make is you want to have this pristine protocol, so you put in lots of inclusion-exclusion criteria, and then the study starts and you can't get patients in your study and you start relaxing your inclusion-exclusion criteria. I think this was mentioned last night in the talk about the number of amendments that you have. That's one of the main reasons that you have amendments, is you want it to be clean, and you put this tight criteria in place that are just impossible to recruit to, and you have to relax that. But it's also important when you think of it in terms of generalizability. At the end of the study, right, who that generalizes to if you've made your restrictions too tight. One of the best examples of that I can give is, again, this goes back to the 90s. I'm old, so all my examples go to the 90s. Except for my music example with Will, that was but so there was a big controversy for ephedra in the 90s, which was like a weight loss drug that was an herbal supplement sort of sold over the clinic. And there were lots of trials because there were cardiac problems that came up with people who took that. But there were lots of trials. It was a really interesting case study in terms of this whole inclusion-exclusion in clinical trials. And that there were questions of whether there were clinical trials that had been done in ephedra to show that it was safe and effective at weight loss versus whether there was a risk. But actually, both were true. There were several published clinical trials in that area that showed, like, you know, if you were severely obese, needed to lose weight, ephedra was effective at doing that, and did not have major safety risk. But at the same time, there were lots of cardiac problems being reported by people taking ephedra. And if you look at if you actually read the papers of the clinical trials, what you'll find is if you were predisposed to cardiac problems, you were not eligible for the clinical trials, right? But once ephedra is like, so I mean, at the time, some of you probably don't even remember this, but at the time you could walk into, you know, Circle K or Cookie Mart or whatever and buy ephedra right there at the counter. So, you know, a lot of people who've taken it are probably those people who are predisposed to cardiac events who could not get into clinical trials. So you had both were true. Is the drug potentially unsafe? Well, probably yes. Was it shown to be safe in clinical trials? Yes, but it was shown to be safe in the population who were in those clinical trials, which are not necessarily the same as the population who was taking the drug. So you have to keep all that in mind when you're defining the population, is how you can generalize it and what those results then apply to. Now, all this leads to really what is the crux of why you're here, which is your study protocol, right? The protocol will guide everything in the trial in terms of what you want to do. So it needs to be kind of a comprehensive description of key information. And then, so many of you have written grants, and there's this whole idea of grantsmanship. There's kind of this whole concept of protocolmanship as well, right? And you know, some of Will, Karen, Bill, some of the others who are here are really good, you know, at doing this. That you want to write a protocol as descriptive and thorough as possible, but it doesn't want to be you don't want to be too precise in writing a protocol. If you're too precise, then every time something has to modify, you've got to make a protocol amendment, and those are a pain. So you want to allow yourself flexibility, but not too much flexibility, right? So it's an artistic endeavor to write a good protocol in terms of doing that. And that's a lot of what you're going to do this week is working on a lot of the main aspects of that, but in putting it all together, that's where kind of the artistic piece gets together. And you'll know you're a real clinical trialist when you're just a complete nerd and you read someone's protocol and you're like, man, that is nice written. So I told my wife, so, you know, this is summer, this is when all the kids are going to camp. When she was driving me to the airport yesterday, I'm like, I thought I'm going to clinical trials camp. Right? Clinical trials camp. Not as much fun as swim camp or gone. But some of the things that should be in the protocol. So I pulled this from, you know, NINDS has a protocol template on their website. So I kind of pulled this. And a lot of the networks, so Neuromex, I think NAD, StrokeNet, are using the same type of template for protocols within the network, which is likely what you guys are using here. But this just covers some of the things that are listed there that need to be addressed. The study objective. So this gets back to what's the question, what's the primary objective, what are the secondary objectives. Background, you know, some rationale, some supporting data for what you're doing. Study design, how do you describe the design of your study? It's randomized, how's it set up, how many groups, how long is follow-up. The selection and enrollment, so this is the inclusion and exclusion. Description of the study medication, the drug, the device, what is it you're studying, how are you getting it? Are there any clinical laboratory evaluations? Those should be described. The management of adverse experience, so that's an important component of the protocol. So this should lay out not just what is an AE, but how they're going to be managed during the study, right? And where that leads to more, not so much on what you're doing, although it could. If you're doing something under an IND, then you have federal reporting requirements that have to be met. So if you have something, say you're looking at a drug under an IND, if a serious adverse event happens that is potentially related to that drug and unexpected, then it has to be reported to the FDA within 7 or 14 days, depending upon some different criteria. But you need to have procedures in place for who would make that determination. So NINDS often has independent medical monitors for studies that would be the size of what you're doing, who would do that. So it would be somebody independent of the study. Because you, as the investigator, you should not be a person doing that, right? If it's your study, your drug that you're studying, you shouldn't be deciding whether it meets the criteria for reporting. You want that to be independent. So it'd be an independent medical monitor, somebody who's not involved in the study, who might receive a notification if this happens. They would review all the information, make a determination, does it appear to be related to the intervention, is it expected or unexpected, and based on their classification, that then determines whether you need to report or not. So again, it's to try to keep the separation away from you so that you're not at risk. Because again, even consciously or subconsciously, it's probably going to weigh into how you evaluate that if you have a vested interest in terms of how the study turns out. Statistical considerations. This is the most important section of the protocol. This is the section that you guys will probably never write. If you're smart, you'll find a good statistician to write this for you. Actually, when I read you guys, I would appreciate it if you would find a good statistician to write this for you. But these are important points, right? The sample size section, how you're going to analyze the data. These are all things again. I will tell you that it is good if you can find a statistician that works with you and understands what you're doing and doesn't just give you boilerplate text to put in there. From a grantsmanship perspective, it is very easy to tell when the statistician and the clinician have maybe never talked to each other in person. There's just a sample size section that was emailed and pasted straight into that, and those stand out like a sore thumb. And you can tell, and you can kind of get a sense just from that section of how integrated the teams are, which does affect, at least what I read, that it affects the score. Because if the statistician and the clinician can't collaborate on the grant application, it's hard to imagine how well they're going to collaborate on the actual clinical trial. So I think it's important to make sure that there's some consistency there in terms of what you're trying to do. Data collection, site monitoring, all that. Human subjects, so that's important. Publication of research findings. If it's a multi-site study or there's multiple investigators, it's often important to lay out how the primary results, how the authorship is going to work on the primary results. I think that's extremely important for you guys because you're junior investigators, and so publications are going to be important for you. What you don't want to end up doing is you're in a situation where you're doing all the work and then you don't get any of the papers at the end because somebody else is coming in and says, well, I'm going to take those. I'll write those papers at the end of the study. So you want to make sure that you get credit for your effort. And it's also, clinical trials are tough in that usually the payoff is at the end. So as a junior investigator, when you're doing a trial, you're not going to get many papers. Maybe there's a design paper at the front, but it's at the end of the trial is when most of the papers will come out. So, you're not going to be you're going to be doing a lot of work and not getting many publications for a big chunk of that time, which is tough when you're a junior investigator and in any type of field in academics, publications are currency, right? So you want to work with that. So you need to be cognizant of that. But that's where areas like if there are existing data sets or other trials that have papers that need to be written, a lot of times at the end there's more papers than there are people to write them. And a lot of trials that have data sets out there are looking for people that can come in and lead writing groups and help move those things through. If there's opportunities to do that, it'll give you good networking experience, it'll give you a chance to get papers, but it'll also give you a sense to learn more about clinical trials just by going through the process of doing that and understanding the things that are coming out of other studies. That's all I have. I'd be glad to take any questions. I won't name any, we'll do that tomorrow.
That was great. So I had a question about your phase 2 to phase 3 transition. If you're like in your phase 2 trial, you get a really small effect size, and you push that smaller in, that's obviously going to change your powering or sort of the things be for your phase 3. Like how are you? Do you just have how are you managing that?
So here's my preference, and then there's reality, right? So I really don't like using effects observed in phase 2 to power phase 3 studies for a couple of reasons. One, it's an estimate. Because they're small sample sizes, as you mentioned, there's often a pretty wide confidence interval. And also, it's typically, there's a lot of literature that shows that they're usually biased in a positive manner. So whatever effect you see is probably larger than the actual effect. Just for a couple of reasons, right? One, you're only going to do the phase 3 if you saw something you liked in phase 2. So almost by definition, it's got to be somewhat on the positive side. So you can end up overestimating your effect. The other thing is it's kind of dangerous to use any type of estimated effect in some sense because you're chasing statistical significance, not necessarily clinical significance, right? So you might see a large effect in a prior study. If you power your study for the large effect, that's what you're powered to address. So from the answering the question standpoint, if you see that large effect, you're going to get a statistically significant result. But maybe a moderate effect, right? Maybe effect smaller than that, is still clinically meaningful, but you're going to miss that in your trial, right? And so that's where I think it's important to kind of have a sense. It's easier in some areas than others. The sense of what size effect do you need to see that would be clinically meaningful, right? Taking the statistics or the estimation out. If I saw this, it would be meaningful to the field. Anything lower than that's probably not going to impact, the ability to treat patients. And so I prefer to try to power studies based on those size effects. If you can't, the estimates help you get into a range. But often I think if you have an estimate, say of like a 30% increase, I usually would like to bump that down some, right, to be somewhat conservative and try to show that you have power over a range that varies somewhat. The other thing that I try to encourage people to do, and it's sometimes tough in the early protocols that you guys are working on, but I always try to encourage people to power for 90% if possible, not 80%. And the reason for that is if you look, like a power curve is this sigmoidal shape, it's kind of this S-shaped curve. And if you're at 90%, you're kind of up here more on the flatter area. So if you're off on your estimates, it's not going to move as much. If you're at 80%, you're more closer to like the steep slope, right, with the curve. So if you're at 80% and you're off of your assumptions, there's a bigger drop that you can see. So if you power at 90%, you're protected somewhat if you've made overly optimistic assumptions in your sample size population parameters, which I don't know what George, but in my experience, like 95% of the time, that's what happens. It's definitely not equally likely in both directions. You want to build in a little bit of protection of these. Is that what you're saying?
So I have a question about your comments regarding phase 2 clinical trials, and you mentioned that you can't really truly test efficacy with, you just don't have the power. So as we kind of develop our objectives, is there just a proper way to state something you're looking for if you're testing a specific drug? Because you can't say efficacy because you're not going to be able to answer that question in phase 2 trial. Yeah.
So let me rephrase that. So you can't show efficacy like on the main clinical endpoint that would be like for an FDA type application. You could show efficacy on like if you had biomarkers, which again like we did have a whole course on biomarkers. If you have a biomarker or a decent surrogate, you could show efficacy on like a surrogate. Maybe there's some imaging metric that you could show efficacy on in phase 2, and then in phase 3 you're looking at the clinical endpoint, which might be a larger sample size. Or there are also designs, I think, let's talk about futility. Valerie, we'll talk about futility designs, which are a way to try to get at that, where you're setting up a threshold that you have to cross to justify going forward to phase 3. So it's not looking, so with the futility design, the question is there enough evidence that you can determine that it's futile to go to phase 3 or it's supported to go to phase 3, but you're not trying to show that it's efficacious. The phase 3 trial will sort it out. And so I think when Valerie explains that, you'll see that. So that's a way that you can do a phase 2 trial targeting a question, but not needing the power for the efficacy, which is decided to be made for phase 3. And there's other approaches like that that you can use to try to get at that.
It was on.
All right. Can you hear me?
Yeah,
okay.
Absolutely.
So one thing to keep in mind when you're designing a phase 2, a lot of times the goal is not necessarily to answer a question definitively there, it's to get information about the phase 3. I mean, I see a ton of studies where they're trying to find phase 2 is a dose finding exercise, but they power the study, can I find an effect? When the real question in the study is, do I know the right dose to take into phase 3? Do I know the right population? So I think it's always worthwhile in a phase 2 program to think, at the end of this study, what do I need to know, and am I going to be in a position to actually do that phase 3 study? So am I going to know the right dose? Which is different than just a standard power calculation. It's what do I need to achieve in this study and make sure that I actually achieve that.
I think I agree with that. I think that this comes up in grants a lot though, is if you're doing a phase 2 study, there needs to be some threshold that you need to meet to go to phase 3, especially in the academic trials. This is where I think academic trials are a little different from industry trials in particular. Because if you're already made your mind up you're going to phase 3, then from an academic trial standpoint, it's hard to justify that in a grant. And that often comes up in study section when you're just doing this to figure out what dose to take to phase 3, because you've already decided you're doing phase 3. That's a bit more problematic. That's more of the industry model, but it doesn't, at least the study section I've been on, that doesn't fly as well.
Can I comment on that too? So I like to think of phase 2, or what I call middle phase, as learning phase. So I totally agree with Kurt, is that this is your learning phase. If you have all the information to go to an efficacy trial, you don't need middle phase, you're already there. Middle phase, or phase 2, is so you can gather the rest of the information. Is it safe? Or is it safe enough? Is it feasible? Can you even pull it off? Can you get the patients in? Can you introduce the treatment? Do people quit too early? You know, all that stuff you have to do, plus dose finding. What's the dose? What's the timing? How long do you give it? All that stuff is in the learning phase, or the middle phase. So that's what I think of. I know a lot of you guys come in and are thinking about efficacy. Does it work? Does it make people better? But you can't start with that. There's all this kind of background stuff that you've got to gather, and that's middle phase, or phase 2. That's how I